All right, in section 22.8, what we're going to do here is talk about what happens once the messenger RNA gets the code. How does it now synthesize the protein from the code into the protein? So now we're going to be talking about the tRNA, all right, or the translation RNA. So the messenger RNA contains a sequence of the codons that determines the amino acids of the protein. Individual tRNAs bring specific amino acids to the peptide chain. Remember that the tRNA was that clover looking thing. I'll try to write it here. It looked like that, all right, and it has the anticodon on the tip, and then it has the particular amino acid that translates for that codon on the other end of it. All right. So the RNA is it contains the binding site in the ribosome for all this to happen. So the messenger RNA gets the codon from the DNA. So the DNA has a sequence, and then you get 3 of these codons for the sequence, and then the tRNA has the anticodon for it, which matches up with the mRNA, and there is the amino acid is hanging off of this, which they line up, and eventually all of the amino acids hook up, or the tRNA is for a particular amino acid, and then all the tRNAs finally bind, and all the amino acids hook up. And that's kind of what this slide is all about. Okay, so each tRNA contains an anticodon of 3 nucleotides that is complementary to the codon in the messenger RNA and identifies the individual amino acids. Ironically, the anticodon in the tRNA is the actual sequence in the DNA, because remember, the mRNA, all right, is the opposite, or it's the base pair of what's in the DNA, and then the tRNA is the opposite of this, so technically this is actually what we have in our DNA. All right, so it's kind of interesting how that works. All right, and then the 3 main parts of translation is initiation, elongation, and termination. Okay, but basically, as I was saying, the tRNA, which is right here, this is our tRNA. All right, there is an anticodon. All right, if we go back, let's go back to the where it shows you a picture of the tRNA. Here is the tRNA. All right, we have the anticodon. This is the opposite letters that's in the mRNA. Okay, and what's going to happen is each of these is going to pair up with the mRNA, and there's a particular amino acid that corresponds to whatever the anticodon is. Right here, it kind of shows you a picture. Here, I believe this site right here is showing you the anticodons, and then this is the amino acid that represents it. Okay, so this is the tRNA. I remember it because it's shaped like a T. That's the T. So I don't think that's why they call it that, but that's how I remember it. They actually call it a cloverleaf, but you can look at it however you want. Okay, so you have the tRNA so the mRNA comes from our DNA and goes into the ribosome, all right, with its codon, its 3 nucleotides, and then the corresponding tRNA that corresponds to the opposite of each of these hooks up, and it has an amino acid associated with it. What's going to happen now is another one of these, all right, is going to come by. Let's put amino acid, and its codons are going to pair up with this one right next to it, and it's going to come in here and hook into here, all right, and you'll have an amino acid that corresponds, and then these eventually will get hooked onto each other, and that will finally come off, and then another one will come in and hook onto there, and then another and hooks on, and another hooks on, and so forth and so on. That's basically how this works. All right, so how the elongation and the termination, how it works, here's kind of a picture. So the initial, the tRNA, it comes in that matches up with what our mRNA has, which again came from our DNA. All right, then a second tRNA will come in, which matches the opposite of what this is. So you have UCA, so the opposite is AGU, so which is for a serine amino acid. That comes in, they bond, all right? So it unhooks from the current, the tRNA, and you now have a bond between these two amino acids. As this slides down, this one will get kicked out. It gets moved into that place, and then you have our next code. In this case, it's GAG, which is CUC for the anticodon right there. Matches up with this one. It goes into place of it, and then the glutamine hooks up with the serine, and then it just keeps going. It just keeps going through, and each of these tRNAs keep adding in, all depending on what our sequence is, on our elongated, in our very long mRNA. So whatever our mRNA has for its codons, the corresponding anticodon pairs up with it, and whatever amino acid is in that hooks onto here. All right, it's actually amazing how they were able to figure all this out. Okay, so the translation continues until you have a stopping codon of either UAA, UAG, or UGA is reached, which is called the termination. All right, and the completion of the protein. Okay, and so here just kind of gives a summary of everything. So you have your DNA strand, the mRNA, all right, it copies the our template here. So the TAC, all right, so remember that it goes with T, but remember there is no T in mRNA. The T is equivalent of U, all right, so in RNA, the T equals U, all right, so this is the messenger RNA, okay, which then gets it gets paired up with the anticodon of the tRNA. Now notice that our tRNA is the same as what was actually in our DNA. Remember, U though is, so if you think of this as a T, TAC, that's what I actually had in my DNA. So it's kind of doing an opposite thing. Again, it's amazing how we have very small amount of errors occurring because there's so many things changing. So the TAC gets coded to the AUG, which then gets paired up with the anticodon, which was actually initially the same initials as our DNA. And then each of those has an amino acid hook to it, and then they all hook up and form the protein. All right. We will definitely talk more about this when we get into class.